Good afternoon. My name is Miguel Espero. He first starts us off by introducing us to Gregor Mendel and the discoveries that he found, and he found the that genes come in pairs, and usually one coming from each parent. And because of that, he was able to track the parental genes and see which ones were the dominant and the recessive genes. And from this, he was able to create the fundamental laws of inheritance. Now he did this through his experiment testing out pea plants, and he did this by looking at the 7 different traits found in pea plants, each having each trait having 2 forms. And he first started off by crossing pure breeds, which created the F1 hybrids. In those F1 hybrids, they all had the same traits, meaning they all had the same color and same appearance. With those F1, he crossed them again and produced the F2 hybrids. This is where he actually saw changes occurring in them. In the F2, only 75% had purple flowers, while the rest were all white flowers. From there, he introduces us to Thomas Morgan, and he created the chromosome theory of heredity, and he was able to confirm Mendelian's laws of inheritance because of that. In his experiment, he studied the traits passed from one generation of Drosophila melanogaster to the other, and he started by crossing wild types which had red eyes together, and it took him 2 years to find the mutant trait of Drosophila melanogaster, which was white eyes. From there, he isolated the white-eyed fly in a jar and filled it with female red eyes to see what the offspring would produce. From this, he learned that it took 2 generations to see the mutant strain appear in the new offspring. And the reason that he used Drosophila melanogaster was because it was easy to put them all in a container because they were so small, so you can have hundreds or thousands of them, and they would produce a number of offspring at the same time. From there, we go on to James Watson and Francis Crick. These were the ones who discovered the chemical structure of DNA, and this is what revolutionized biology and medicine because we now knew what the structure was. And they did this by X-ray crystallography to show the backbones. Rosalind Franklin is someone who helped them find the structure, and she was well known for her X-ray crystallography, and the most well known of her pictures was number 51 because it was the one that clearly showed the double helix structure of DNA. But sadly, she was not able to get the Nobel Prize because she died 4 years before Watson and Crick were given the Nobel Prize. Now, Linus Pauling is someone who was also trying to figure out the structure of DNA and was actually able to publish a paper before Watson and Crick. But in that paper, he said that the glue that held together DNA was magnesium, and Watson and Crick didn't think that was right, so they went around asking people who knew chemistry, and they were able to figure out that if DNA actually did have magnesium bonds, the bond would be way too weak and it would come apart easily. And this is what made it so that Linus Pauling wasn't able to get the credit for the DNA structure, while Watson and Crick did. Now, Boyer and Swanson, they're They were able to pioneer the field of recombinant DNA technology and biotechnology, and they were able to find a way to make bacteria make proteins, which also helped to make which allowed genetic engineering to get a head start. And in the future, with newer technology, better technology, and more advanced knowledge of genes and how to use them to help us, I think that we'd be able to find better cures for genetic disorders, and that with that new technology, we'd be able to pinpoint the genes that are affecting someone and then treat them through better methods of gene therapy. Now, on the topic of gene mutations and human diseases, Archibald Giroud was one of the first to discover that diseases could be passed down through families because um, he noticed that a newborn child um, had gotten the same disease that someone else in the family had. Because of this, he was able to figure out that some factors were inherited and altered the newborn's genes. And in 1998, McCusick found that there were more than 12,000 variations in genes that could be linked to traits and disorders that could be mild or life-threatening. And he found that mutations in a single gene can cause different effects on a wide variety of organs, such like connective tissue, like tendons, cartilage, bones, ligaments, and it could lead to problems like cardiovascular manifestations, and mutations in structural proteins could lead to heart failure and aortic ruptures, like the ones found in Morphin disease, where the person tends to die young from ruptured blood vessels. Now, eugenics is a topic that causes a lot of controversy because um, many believe it was bastardized by the Nazis as a way to get rid of the Jews and make themselves as superior. And I just think it's something that shouldn't be considered um, for thinking of future generations because we don't really have a right to choose who can reproduce and make offspring, and it's just something that shouldn't be touched. Cloning, on the other hand, is a separate issue all on its own because it comes with its own set of issues. There's cloning complete organisms, as you can see here, where they clone a pig or a dolly with sheep, where it could be used to save a species from going extinct. But a lot of people think of it as bringing back a pet that has died or bringing back someone who they loved. But the problem with this is that they fail to realize that it wouldn't be a carbon copy of that person or pet. Um, but in terms of therapeutic cloning, I feel that it's viable because its only use is to repair or replace damaged tissue and organs. Now, racial categorizing is something that we all go that has still goes through the world today. But a lot of genetic research has shown that there's no basis for one race being superior to another because not enough time has passed. And not only that, all humans originated from the African continent 100,000 years ago, and genetic variation takes millions of years to actually occur. A little bit about the author's family. There is mental health issues on his father's side, and his father does suffer from NPH. NPH is normal pressure hydrocephalus. It's a genetic disease that the person can trigger from the age of 5 to and 80 years old. His father triggered it when he fell off of a rocking chair, and it's a result from abnormal fluid flow around the brain. It can cause dementia, gait instability, and urinary incontinence. Continuance.